Okay, thank you. I'm going to talk about fishing for a cure. So I do have to do, I think, a brief introduction to Batten disease. It was first identified by Frederick Batten, that's why it's called Batten disease. He was a neurologist in 1903, and what he really noticed was the marked reduced life expectancy in patients that he saw with this. It's a rare disease. They're simple genetic diseases, and this is a schematic taken from the leaflet we produce for families. If you're an unaffected carrier, you won't know this. It's a single, different mutations, but in single genes. There's actually 14 been identified for Batten disease, so you won't realise this. And then obviously the risk for each pregnancy, if you're a carrier, there's a 1 in 4 chance that the child could be affected. And we do have, some families suddenly where 2 or 3 children are affected before they realise that they're carriers. So, and the one I'm going to talk about today is CLN2 disease. For many of them, we don't even know what the proteins do. We've identified the genes, but not the proteins. For CLN2 disease, we do. An enzyme is missing, so you have a single defect in your gene, and therefore that missing, you don't produce the enzyme, and this leads to disease. And it is a devastating disease. These are lysosomal proteins. Lysosomes are where break down and recycling of waste products. And really, I'm going to cut to the chase here. The damage is neuronal, it's neurodegenerative. For these children, life expectancy, depending on the type, can be between 6 and 30. For CLN2, it's probably between 6 and 12. And they will have seizures. They will lose their ability to walk, talk, eat. They'll become completely dependent on their parents. It is a devastating disease. Death at the moment is inevitable. And I'd like to introduce you to a very special person, and I might just like to ask for 10 extra seconds because I think we just need to look at this. This is Katie Freeman. Sadly, she passed away when she was 6 years old. She had classic late infantile CLN2 disease. And now, on a happy point, I'd like to introduce you to 3 very special people. So one side is her parents, Karen and Martin Freeman, and they, in memory of Katie and their legacy for her, they wanted to raise money for research. And to the right is Claire Russell, who's also here today, and she's the researcher that we're going to talk about research. Where do the BDFA, where do we fit in? Well, we provide support for families. You can imagine by what I've described to you, support is very, very essential. We raise awareness, and we also fund research. We have our own research project, but we partner with Sparks. It's all peer-reviewed as well, and I really don't have time to go into that. But essentially, we also identify areas in which key things need to be done, and often in rare disease research, to get things going, it is the family groups, it is or individual families. So that's raising money that starts often what we call proof of concept studies. So they wanted to fund this in CLN2 research, and people had already said to us, you need to look at zebrafish. Why use zebrafish? Well, because you can generate lots of zebrafish. The larvae that they produce, you can see them, they're clear, you can do great studies on them, and they would be ideal for screening. You put 2 zebrafish together, and as Claire will tell you, lighting is also important. You will get a lot of zebrafish, so a lot of larvae. That's true. That's one thing we learned from lab day. So they're brilliant for that. And you can also do mechanistic studies, which I do think actually in rare diseases that's important as well when you don't know what's going on. So Claire had a very talented PhD student, and I think you can just see from this visually, you can see from the wild type to the severe CLN2 phenotype, that can you produce a model that really reproduces the key aspects of the phenotype that will be valuable for screening? Is this going to give us some meaningful data? And again, then can you develop a screen? This is some of the first work that was done, and you can see zebrafish, the zebrafish model that they generated. It shows seizures early on, goodness, and so you could model seizure-like activity, movement is different. As I think you can see from this clearly, if you can produce from this, you can screen drugs. So you also need the equipment, automation, and this is what the funding from the Freeman family went for. It's for providing a machine, and also the expertise. This is Ginny, who worked in Claire's lab, and she did most of this work. So this is the screen. Obviously you take an FDA approved library to start off with, because this is what we've all been talking about, and looking at seizure activity and movement. And they screened 500, and actually from the screen, sorry I missed the number of compounds, from 560, 90 were retested, and we have one, Claire has one promising lead. Sorry, no, yes. So, and you can see that we have, her initial results are that you've got an improvement in your seizure activity, but greater movement, and most importantly perhaps increased survival. So as we've talked about, we've heard a lot, really, where do we go from here? And this is what the BDFA are working for very much with the RBC, with Claire, and with the Freeman family. And I suppose those in blue are those of us that are already on board doing this. And we know that we need commercial partners. This is one of the trickiest questions that we had to move on, and clinicians and families involved in this. So it's just a summary of, again, the main people involved. And I think I really have to finish, which is not bad. These are the families. These are all families, also children affected this year, and we have a conference every year. And thank you very much.